Hi, my name is Jennifer Zoe, and I'm a PhD student at UCLA in the labs of Jason Ernst and Eliezer Eskin. Today I'll be talking about identifying global patterns of epigenetic variation across individuals. Understanding molecular variation across individuals is fundamental to understanding variation in complex traits. Recently, molecular data has been collected across many individuals, which has allowed for a more comprehensive identification of variation. One common way to identify molecular variation is through QTL analysis. In this type of analysis, a molecular phenotype is collected in multiple individuals and correlated with a genetic variant. An association test is performed to identify variants that are significantly associated with the molecular phenotype. A similar approach is to do differential peak analysis. This analysis identifies regions that are variable between groups of individuals. In a paper published by the Geschwind and Prabhakar labs, they collected histomodification data in both autism cases and controls and were able to identify many peaks that were differential between the two groups. While the previous two methods did separate analyses for each type of data, another approach is to do a joint analysis across multiple data types. One way to do this is through chromatin state analysis. In a paper from the Snyder lab published in 2013, they used the RegSys software to identify combinatorial patterns of histone modifications and CTCF. In this analysis, the model was trained by virtually concatenating all 19 individuals for each mark, so that the emission matrix has one row for each state and one column for each type of data. After running the parameters for the model, the genomes of the 19 individuals were segmented according to the patterns identified using only the individual-specific data. On the right, each track is a genome segmentation for one individual, and the colors in the segmentations correspond to one of the states learned in the model. The region boxed in red is a variable region across individuals, and the region boxed in blue is a non-variable region. Although these approaches identify variable regions of the genome, none of these approaches identify patterns across individuals that reoccur in the genome. They all look for variation locally in a specific consensus region. One reason that we might expect to observe reoccurring patterns of epigenetic variation across individuals is trans-regulation. Differential expression of transcription factors can potentially affect the transcription of many other genes in the genome. Since histone modifications are known to impact local gene expression, we may expect to observe similar patterns of epigenetic marks across individuals in regions that are regulated by the same transcription factors. The goals of this project are, first, to identify if these global patterns exist, and second, to see how related they are to other types of variation. To learn these recurring patterns across individuals, we propose to apply an alternate stacked version of the RegSys framework. In this framework, each mark from each individual is conditionally independent given the hidden state. In addition to allowing for joint analysis across marks in individuals, the HMM framework takes into account the spatial context of the data. While other methods rely on identifying a set of consensus regions across all individuals, the input to this method is the genome-wide histone modification signal. In this framework, each hidden state is a global pattern across both marks in individuals, and each cell of the emission matrix corresponds to the probability of observing a mark in a specific individual given the hidden state. For instance, S2 corresponds to a constitutive state where all individuals are likely to have signal in all marks, so the emission probabilities for S2 are all high. S3 corresponds to a constitutive state where none of the individuals are likely to have signal in any of the marks, so the emission probabilities for S3 are all low. In practice, we're more interested in the non-constitutive states where signal is different across individuals. In this toy example, S1 has a pattern of variation across individuals and marks that is learned in the emission probabilities. We applied this framework to a previously published dataset of 75 individuals in the lymphoblastoid cell line. In this study, they collected 3 histone modifications that are associated with promoters and enhancers, and they used matched RNA-seq data for 54 individuals. To generate the input to the HMM, we first quantify the number of reads in 200 base pair non-overlapping bins of the genome. We then use quasi-Poisson regression to correct for the effects of known covariates. We introduce this correction step in order to reduce the effect of technical covariates and increase the number of global patterns associated with biological signal. The plot on the right shows an example of how the correlation between the signal and the number of reads in a library gets closer to 0 after applying this correction. The correlations are computed across individuals, and each line shows the distribution of the correlations over bins in the genome. After the correction step, we binarize the signal using a Poisson background model, similar to the traditional CROM-HMM framework. Using the binarized data, we learn a joint HMM model across all individuals and marks. The emission matrix shown on the right is grouped by mark. Within each mark, the ordering of individuals is the same. Within each state, there are similarities in the emission parameters between the marks, which matches our prior knowledge about these marks. This correlation was learned in our model, even though the model is agnostic to the mark labels. We computed the correlation between each pair of marks within each state. We show that the median correlation across states is high, and that the median correlation is maximized using a 90-state model, which we use for subsequent analysis. While this analysis does not rule out confounding factors between samples, the consistency across different histone modifications suggests that the global patterns may be biological rather than technical. We also show that the global patterns are associated with external data sets, such as gene expression. Using the global patterns we learned, we segmented the genome and assigned each 200 base pair window to one of the states. Then, for each gene, we correlated the expression of the gene with the emission parameters from each state assignment within 500 kb from the transcription start site. The heat map on the right shows average correlations across all genes. We observe higher correlation in non-constitutive states near the TSS of genes. This shows that the global patterns near transcription start sites are associated with the expression of many genes. One application of this framework is to predict trans-regulators using the genome segmentation outputted by our framework. We identified predicted trans-regulators using two criteria. First, we computed motif enrichments for the 90-state model using the genome segmentation. We adjusted the p-values for multiple testing, and we additionally required that the motifs have at least a log 2-fold enrichment of 1.5. Then we correlated the expression of each of these transcription factors and identified those that were significantly associated with the global patterns at an FDR of 5%. Transcription factors that fall into both of these categories have a strong potential to act as trans-regulators because they have a potential mechanism by which they are regulated and affect other genes. We identified 18 genes meeting these criteria, and the heat map in red shows the log 2-fold enrichments for the states in which the motifs are significant. Now we'll take a closer look at one of the predicted trans-regulators, REL, that I've highlighted in the heat map. REL is a subunit of NF-kappa B that mediates sequence-specific binding. NF-kappa B is a family of dimeric transcription factors that are known to act as trans-regulators in B cells, from which the lymphoblastoid cell line is derived. In addition to the REL motif being enriched in different regions of the genome, the REL protein is differentially expressed, and the expression levels strongly correlate with state 37 in our model. Thus, the differential expression of REL can potentially affect many other genes. In order to demonstrate that this framework is useful for samples from primary tissues, and to see whether global patterns are associated with autism disease status, we applied this framework to a dataset of autism cases and controls in the prefrontal cortex tissue. We trained models with numbers of hidden states ranging from 5 to 100. We selected the 90-state model, which maximized the number of states associated with disease status after correcting for multiple testing of states and models. In this plot, I'm showing the emission parameters learned for the 90-state model, and on the y-axis, I've annotated whether each sample is a case or control. And on the x-axis, I've annotated whether each state is significantly associated with the disease status or not, where the dark red states are significantly associated. To more quantitatively visualize the associations of the missions with the disease status, I've plotted two box plots showing the diagnosis status on the x-axis and the missions on the y-axis. We applied the same framework as the LCL dataset to identify putative trans-regulators that have both enriched motifs in the segmentation and expression matching the global patterns. We identified 16 transcription factors matching these criteria, and moreover, 12 of these predicted trans-regulators are enriched in 2 of the states associated with disease status, shown here. In summary, we've learned global epigenetic patterns across individuals in both LCL data and autism data using a multivariate HMM. We show that the global patterns are associated with external biological data sets such as gene expression and transcription factor binding, and that they can be used to identify potential trans-regulators. Finally, we showed that the autism global patterns are associated with disease status and that a number of the predicted trans-regulators have motif enrichments in these states. I would like to acknowledge my labs, particularly my advisor, Jason. I would also like to acknowledge our collaborators, Dan and Gokul, who provided the autism data and some valuable feedback. Also, thanks to the conference organizers for the opportunity to present my work and our funding sources. Thank you.